Okay, so while it's true that there's a boatload of commonalities, universal homologies that hold all of known life together, the two domains of prokaryotic life, archaea and bacteria, have some pretty major differences, and this suggests a very early split. Some recent models actually place their split before the establishment of cellular membranes. Russell and Martin, 2001, say that the ancestral cells were encased in rock membranes, and the evolution of bilipid membranes took place independently for the archaea and the bacteria as they left the rocky compartments. This would explain the fundamental difference between the lipids in the membranes of these two domains. Now, the recognition of archaea and bacteria as distinct structures, as distinct domains, this is a relatively recent thing. It was in the 1970s that Carl Wiese first acknowledged their special status and classified the two groups as archaea as distinct from the eubacteria, terms synonymous with archaea and bacteria. The rationale for distinguishing the archaea from the bacteria were molecular differences that were only just then being detected based on the emergence of new technologies. Up until then, the fact that they were prokaryotic, that is, lacking a membrane-bound nucleus, made them appear to be part of the bacteria. In the time since Wiese, we've added more organisms of the archaea. All of them are unicellular and prokaryotic, and the most well-characterized of them live in exotic and/or extreme habitats. We also now recognize that archaea are very common and sometimes important players in ecosystems, even though they lack the high profile. They don't get the attention like the organisms that are either larger or having some more direct impact on the human condition. The other way in which the archaea are central to our BIO202 narrative is the role that they play in the origin of the eukarya. These are the cells that became the first cell with a nucleus, and bacterial organisms being the endosymbionts that formed typical eukaryotic structures like mitochondria. Wiese, in his original description of archaeal differences from bacteria, pointed out that some of these archaeal characteristics followed patterns that actually made them look more similar to our own cells. And this started us along a path toward our current understanding of the origin of eukaryotic cells from some type of dawn cell, an eocyte, that was an archaeal organism. This cladogram from the textbook shows the eukarya as a basal offshoot from before the split between the Euryarchaeota and the TACK supergroup. Some new findings might put a change here. Cells dredged up from the Loki's Castle hydrothermal vent site in the Arctic Ocean have a greater number of molecular commonalities with eukaryotic cells than other archaea, and they're also more closely related to TACK than to the Euryarchaeota. Here's a proposed revision of the relationships from Wikipedia. Findings like these are obviously very exciting, but they revolve around molecular characteristics that aren't so meaningful to a casual learner. I'm going to take a traditional tack here and describe some of those sexier archaeal clays that have some neat and memorable features. Within the Euryarchaeota, there are two groups that are memorably unique. The first group is the methanogens, producers of methane gas. Methane is CH4, the natural gas that we burn for fuel in gas ranges. On first blush, it seems thermodynamically impossible to produce such a high energy molecule as the end product of metabolism. Metabolic pathways are supposed to start with a food molecule with a higher energy potential and end with a waste molecule that has lower energy potential. Clearly, from our experience in using methane as fuel, it has high energy potential, so it doesn't make sense for a metabolic pathway that is set up for generating ATP energy to end up with an explosive molecule like CH4 at the end. You can think about this for a while, and then you realize that CH4 is energy rich in an atmosphere that has available O2 to oxidize the methane gas to produce carbon dioxide. As you might remember from the previous video, methane was a significant fixture in the atmosphere, the main form of atmospheric carbon, until the Proterozoic, when O2 made it no longer stable. Without O2, methane is not especially energy rich, and methanogens are strict anaerobes, living under conditions that are devoid of oxygen. Not so mysterious at all. The metabolism of methanogens is always coupled with that of anaerobic bacteria, that is, from the domain bacteria, that produce fermentive end products like H2, hydrogen gas, acetic acid, formic acid, and methanol. The methanogens pick up these waste molecules from the fermentive bacteria and metabolize them into methane and CO2. Where do you find methanogens? Well, predictably, it's in anaerobic locations that have a large influx of nutrients, nutrients that feed the fermentive bacteria that in turn feed the methanogens. One example is in the digestive tract of cows. Here's a pic of one of my former students. She has now completed her DVM and is a practicing veterinarian. With her doubly gloved arm, she's reaching into the rumen of a cow that's been outfitted with a fistula, which is a connection between two spaces here, between the inside of the cow's rumen and the outside world. A setup like this is needed to study what goes on in the rumen microbial community in situ in the original location. Cows put a whole lot of food into this anaerobic rumen, and the fermentive bacteria and methanogens do their thing, and voila! Lots of methane is produced and released into the atmosphere every time the cow burps and or farts. Methane, as you remember, is a greenhouse gas, a strong greenhouse gas, and the world's cattle population contributes to global warming in a real and measurable way. Another place where you'll find this microbial methanogenic teamwork is the anaerobic digesters at sewage treatment facilities. The influx of food is actually the solid wastes received in raw sewage, to which they add the biomass of microbes that grow fat on the suspended dissolved nutrients that's harvested from the liquid flow through the plant. These solids, poop and microbes, are pumped into the anaerobic digesters, the concrete cylinders that you see from the freeway, and the methane produced from the bacteria and the methanogens is captured and then used in on-plant steam-driven generators to produce the electricity. It's actually pretty amazing, and you should go at some point to witness this in action by taking a tour at your local wastewater treatment plant. The second memorable group of euryarchaeotes is the extreme halophiles, lovers of salt. These cells are the ones featured in the opening page of the textbook chapter on this topic, the one with an extremely salty lagoon that blooms red with halophiles. Halophiles are aerobic and photosynthetic, though not capable of oxygenic photosynthesis. They harvest light energy for the production of ATP, but they don't split water molecules to get electrons. Their red pigments are basically their light capture devices. These cells can live only under extremely high salinities. Ocean water is not nearly salty enough. Okay. Now, there are a lot of pink salts currently popular among foodies that get their color from halophiles. Their pigments have not been washed away from the salt crystals collected from a hypersaline location, like a lagoon that they flood with seawater and allow to evaporate to concentrate the sea salt. This would basically be an ideal habitat for halophiles. Now, here's a bit of biological trivia to support our institutional goal of promoting cultural competency. That is, including awareness of things outside the scope of most students. For me, in my classes, this usually takes the form of food-related issues. It's not real common for me to have students with any real familiarity with a product called salt cod. Bacalhau, if you're Italian, bacalhau, if you're Portuguese. This is a long storage protein product that was really important in the days before refrigeration and is still used in many cuisines. Its preparation starts off with a fresh cod fillet that's packed in salt and then air dried until it has practically no moisture and is so salty that no spoilage microbes can grow on it. Well, there is one microbe that isn't bothered by extreme saltiness. And so it happens that you can occasionally get a red spot on your bacalhau that is perfectly harmless to eat. And this is relevant to our current discussion because I'm going to let you figure the answer to that one out on your own. Now, jumping out of the Euryarchaeota and into the TAC supergroup, and specifically the Crenarchaeota, the C in TAC. This group is memorable because of how some of its members, the thermoacidophiles, require living conditions that are extremely hot and or extremely acidic. Thermoacidophile actually means lovers of heat and acid. These are the cells you can find in places like the hot sulfur springs of Yellowstone National Park, where boiling hot sulfuric acid bubbles up from the bowels of the earth. Now, you don't expect there to be living cells in pools of hot acid, but there are. And up until relatively recently, these cells were the ones that bore the closest resemblance to eukaryotes. And it was speculated that the eocyte, eo for dawn, cyte for cell, this is the hypothesized progenitor of all eukaryotes. It was speculated that the eocyte came from this group of green archaeotes. More recently, however, that closest to us title has moved over to the Loki archaeotes, the ones that were recently dredged up from Arctic Ocean vent communities. Does anyone remember why it took so long for us to discover these archaeal cells? Well, it's because they are low-key archaeotes. Now, one way in which extreme thermophiles, and this includes both archaeal as well as bacterial forms, have turned out to be useful to us is with their enzymes, which are proteins tuned to operate best at very, very high temperatures. In particular, the enzymes used to replicate DNA samples with a polymerase chain reaction, PCR, makes use of some of these enzymes. Now, there's a tendency for people to associate the archaea generally with these highly memorable kinds of organisms, methanogens, salt lovers, hot acid lovers, while the truth is that these are just the exotic outliers compared to the vast majority of the archaea that aren't especially notable or having any direct usefulness for humans. In the oceans, archaea are major components of the microbial biomass, both in the water column as picoplankton, as well as in ocean sediments. Considering the existence of archaeal cells in unknown densities in oceanic crust, it's actually conceivable that the total biomass of archaea on the planet may be greater than the bacteria, or the eukarya for that matter. They just aren't very memorable like the ones we've named in our survey.